Welcome back to Anatomiqa. In this session, we will be discussing the development of the nervous system. The learning objectives of this session are: describe the formation of the neural tube, enumerate the structures developing from the neural crest cells, name the parts of the neural tube and their derivatives, explain briefly the development of different parts of the central nervous system. The nervous system is made of three main parts. The first part is the central nervous system, which includes brain and spinal cord. The second part is the peripheral nervous system, which includes aggregation of cell bodies outside the central nervous system called ganglia, and the nerves that connect the brain and the spinal cord with the peripheral structures. The nerves arising from the base of the brain are called the cranial nerves, and those arising from the spinal cord are called the spinal nerves. Autonomic nervous system is present in both central nervous system and peripheral nervous system, consists of neurons that innervate smooth muscle, cardiac muscle, glandular epithelium, or combinations of these tissues. Please note this important point. All the structures in the nervous system are developing simultaneously, but for our convenience and understanding, we will discuss it one by one. Before going into further development of the neural tube, let us recall the process of neurulation. By the third week of embryonic development, the three definitive primary germ layers are formed in the embryonic plate or the disc through the process of gastrulation. From dorsal to ventral surface of the embryonic disc, the three germ layers are ectoderm, mesoderm, and endoderm. Of these three, only ectoderm will take part in the development of nervous system. We know on the dorsal surface of the embryo, the ectoderm thickens to form the neural plate, which will develop into the neural tube. The process is called the neurulation. The neural tube gives rise to the central nervous system. The neural crest cells, which are formed during the neurulation, gives rise to the cells that form most of the peripheral nervous system and autonomic nervous system. Let us recall the formation of the neural tube little bit in brief. Neural plate and neural tube. As the notochord develops by the end of third week, it induces the ectoderm over it in the median plane to thicken and form an elongated plate of neuroepithelial cells. This thickened plate of neuroepithelial cells are called neural plate. At first, the elongated neural plate corresponds precisely in length to the underlying notochord up to the buccopharyngeal membrane. It appears cranial to the primitive node and dorsal to the notochord and the mesoderm. Gradually, the neural plate enlarges, overgrows the buccopharyngeal membrane and becomes slipper-shaped structure. The cranial end becomes broader and the caudal end remains narrow. Now, over the neural plate, in the center, a groove is formed at approximately 18th day of gestation called neural groove. The ridges bordering the groove raise and grow into neural folds. The lateral edges of the neural folds are continuous with the ectoderm. The neural folds start approximating each other just before fusion of the neural folds, and area of cells on the crest of each of the neural folds get separated from the ectoderm. These are called neural crest cells. They form islands of cells within the mesoderm and surrounds the neural tube. As the neural crest cells are getting separated from the neural folds, the neural fold, which was continuous, now splits into two parts. The two edges ventral to the neural crest cells fuse to form the neural tube, and the ectoderm is now called neurectoderm. The two edges on the dorsal side of the neural crest cells fuse with each other and forms the surface ectoderm. This whole process, starting from neural plate to the formation of the neural tube, is called as neurulation. Neurectoderm and surface ectoderm, which are formed, now differ in anatomic location and in differentiation potentials. The neural tube starts closing from the cervical region. The neural tube closing extends on either side from the cervical region, cranially and caudally, until only small area remains open at both the ends. The cranial opening is called the anterior or cranial or rostral neuropore, and the caudal opening is called posterior or caudal neuropore. The anterior neuropore closes on day 25 and the posterior neuropore closes on day 27. The lumen of the neural tube is now called the neural canal. The neural canal now communicates from both ends with amniotic cavity until the two ends are closed. The walls of the neural tube thickens to form the brain and spinal cord in relation to the fourth pair of somae. The neural tube cranial to fourth pair of somites forms the brain. The neural tube caudal to fourth pair of somites develops into the spinal cord. Neural canal related to the developing brain gives rise to the ventricular system of the brain. The neural canal related to the developing spinal cord gives rise to the central canal of the spinal cord. In this slide, you can observe the diagrammatic and electron microscopic picture of the neural tube and the somites. The surface ectoderm has been reflected in the midline to show the neural tube and the somites, which is very well appreciated in the picture. Let us first discuss on the derivatives of the neural crest cells, and later we will go to development of brain. We know neural crest cells originate from the neural tube. These cells migrate to different parts of embryonic tissues. Neural crest cells are unique to vertebrates and arise from the embryonic neurectoderm of the ectodermal germ layer. Neural crest cells are transient stem cells that give rise to a variety of cell types. Neural crest cells in the cranial region develop into components of eye, dermal bones of the neurocranium or skull, pharyngeal arch cartilage derivatives, that is skeletal and connective tissue components, odontoblasts which form the dentine of teeth, dermis and hypodermis of head and neck, leptomeninges, that is pia and arachnoid matter, parafollicular cells of thyroid gland, aorticopulmonary septum which separates the aorta and pulmonary trunk, sensory ganglia or cranial nerve ganglia of cranial nerve 5, 7, 9 and 10, ciliary ganglia of cranial nerve 3, pterygopalatine ganglia of cranial nerve 7, submandibular ganglia of cranial nerve 7, otic ganglia of cranial nerve 9, and postganglionic cells of parasympathetic ganglia. Neural crest cells in the trunk region develops into melanocytes, Schwann cells of all peripheral nerves, chromaffin cells of adrenal medulla, dorsal root ganglia, postganglionic cells of sympathetic chain ganglia, enteric parasympathetic ganglia of the gut, that is Meissner and Orbach's plexus of cranial nerve 10, and pre-aortic or postganglionic cells of parasympathetic ganglia in the abdominal and pelvic cavity. Neural crest development is important to understand because mistakes in the development can lead to birth defects and cancers. These birth defects and cancers arising from neural crest cells are known as neurocrestopathies and include craniofacial malformations, familial dysautonomia, syndromes like Tracher-Collins, Waardenburg, DiGeorge, and Charge syndromes. By now, we know in the beginning of the development, the neural tube forms a single straight tube. During 4th week of development, as the cranial fold is occurring in the embryonic disc, the brain, which is growing rapidly, also flexes along with it. This bending produces the flexures in the midbrain region, which is called midbrain flexure or cephalic flexure. At the junction of the rhombencephalon and the spinal cord, there is one more flexure which is formed. This is called the cervical flexure. One more flexure develops in the opposite direction between the cephalic and the cervical flexures. This is called the pontine flexure. The formation of the pontine flexure thins the roof of the hindbrain. Now let us discuss the development of each part of the central nervous system. Going from the caudal part of the neural tube, we will first discuss with the spinal cord. The part of the neural tube caudal to 4th pair of somites develops into spinal cord. The neural canal develops into the central canal. We will study the histogenesis in the wall of the spinal cord later in this session. Positional changes of the spinal cord. We should know the varying lengths of the spinal cord from the intrauterine life up to the adult stage of the individual. During the 8th week of development, the spinal cord extends the whole length of the vertebral canal, that is, till the 4th coccygeal vertebra. The spinal nerves pass through the intervertebral foramina opposite their levels of origin. As the development proceeds, the positional relationship between the spinal nerves and the intervertebral foramina change and does not remain the same because of differential growth. The vertebral column and the dura mater grows rapidly compared to the slow growth of the spinal cord. By 24 weeks, the spinal cord comes to lie at the first sacral vertebra. At birth, the lower end of the spinal cord extends to the level of the 3rd lumbar vertebra. Now, after the birth, the lower end of the spinal cord is called conus medullaris. In adults, the conus medullaris terminates at the junction of the 1st and 2nd lumbar vertebra. As a result of this differential growth, the spinal nerves from the lumbar and sacral segments run obliquely downwards from the spinal cord to the corresponding level of intervertebral foramina. Below the level of the spinal cord, the nerves form a bundle called cauda equina, which means horse's tail. The dura and arachnoid matter end at the second sacral vertebra, but the pia mater forms a long thread-like structure called the filum terminale, which extends up to first coccygeal vertebra. The neural tube, cranial to the fourth somite, develops into brain. Even before the anterior neuropore is completely fused, it expands into 3 dilatations called primary brain vesicles. From cranial to caudal, they are prosencephalon, mesencephalon, and rhombencephalon. The primary brain vesicles further divides into secondary brain vesicles. Prosencephalon or forebrain further divides into telencephalon and diencephalon. The second dilatation is the mesencephalon, does not further divide. It remains the same as mesencephalon. And the third dilatation, the rhombencephalon or hindbrain, further divides into metencephalon and myelencephalon. Now the 3 brain vesicles, primitive brain vesicles, after division results in 5 brain vesicles, secondary brain vesicles. The telencephalon develops into cerebrum. Compared to the rest of the neural tube, the telencephalon grows rapidly on all the sides over the diencephalon, which gets buried inside the telencephalon. The diencephalon develops into thalamus, hypothalamus, subthalamus, epithalamus, retina, ciliary body, and iris. The second dilatation, mesencephalon, gives rise to the midbrain. The metencephalon of the rhombencephalon or hindbrain develops into pons ventrally and cerebellum dorsally, and myelencephalon develops into medulla oblongata. In this slide, you can observe the diencephalon completely buried inside the developing telencephalon, that is cerebrum. In this slide, you can observe the diencephalon developing into different parts: epithalamus, thalamus, subthalamus, hypothalamus, posterior lobe of pituitary gland, and retina, ciliary body, and iris. Now these are the parts which are developing from the prosencephalon. Now let us see the histogenesis in general occurring in the wall of the neural tube. When we observe the cross section of the neural tube, the wall of the initial neural tube differentiates itself into 3 zones from the lumen to outside: ventricular zone, intermediate or mantle zone, and marginal zone. Ventricular zone. Initially, the wall of early neural tube consists of neurectoderm, which is composed of pseudostratified columnar neuroepithelium. These cells constitute the ventricular zone. These cells give rise to all the neurons and macroglial cells. A first wave of proliferation and differentiation of the neurectoderm gives rise to neuroblasts, which migrate into the intermediate zone. A second wave of proliferation and differentiation of the neuroectoderm gives rise to glioblasts, which migrate into the intermediate zone and the marginal zone. The neuroectoderm that remains in the ventricular zone after completing its differentiation gives rise to ependymocytes or ependymal cells, tanycytes, and choroid plexus cells. Intermediate or mantle zone. The intermediate zone or the mantle zone contains neuroblasts and glioblasts derived from the pseudostratified columnar neuroepithelium of the ventricular zone. The neuroblasts differentiate into neurons with dendrites and axons. The glioblasts migrated into the intermediate zone differentiates into astrocytes and oligodendrocytes. Hence, the mantle layer develops into the gray matter of the central nervous system. The intermediate zone is divided into two regions. The alar plate or sensory plate, which gives rise to neurons associated with sensory or afferent functions, and the basal plate or motor plate, which gives rise to the neurons associated with motor or efferent functions. Marginal zone. The marginal zone contains the glioblasts derived from pseudostratified columnar neuroepithelium of ventricular zone and axons arising from the neurons present within the intermediate or mantle zone. The glioblasts differentiate into astrocytes and oligodendrocytes. The marginal zone forms the white matter of the central nervous system. Till now, we discussed the histogenesis in general. Let us now discuss the histogenesis occurring in each part of the central nervous system. Going from the caudal part of the neural tube, we will first discuss histogenesis of the spinal cord. The neuroepithelial cells or neurons in the intermediate zone proliferate and differentiate in the lateral wall of the neural tube, forming a thick lateral wall and a thin roof and floor plates. Because of this differential thickening, a shallow longitudinal groove is formed on the lateral wall by the ependymal cells or ependyma. This shallow groove is called sulcus limitans, which separates the dorsal part from the ventral part and is related to the central canal. The sulcus limitans disappears in the adult spinal cord but is retained in the rhomboid fossa of the brainstem. The sulcus limitans extends from the spinal cord to the rostral midbrain. The dorsal part is called the alar plate, which is sensory in nature, and the ventral part is called the basal plate, which is motor in nature. The alar and basal plates produce longitudinal bulges extending through most of the developing spinal cord. Surrounding this layer is the marginal zone, which develops into white matter of the spinal cord. The roof plate is the non-neural roof of the central canal, which connects the two alar plates. The floor plate is the non-neural floor of the central canal, which connects the two basal plates. The floor plate contains the ventral white commissure. The developing spinal cord encloses the central canal lined by ependyma. Basal plate. The basal plate is a ventrolateral thickening of the intermediate zone of the neural tube. The basal plate gives rise to the motor neuroblasts forming the ventral and lateral horns of the spinal cord, which represents the general somatic efferent and the general visceral efferent cell regions. The axons, which arise from motor neuroblasts in the basal plate, exiting the spinal cord, forms the ventral or motor roots. As the basal plates enlarge, they bulge ventrally on each side of the median plane, and ventral medial septum is formed with a longitudinal groove called ventral median fissure. Alar plate. The alar plate is a dorsolateral thickening of the intermediate zone of the neural tube. The alar plate gives rise to sensory neuroblasts of the dorsal horn or dorsal gray column, which presents the general somatic afferent and general visceral afferent cell regions. The alar plate receives axons from the dorsal root ganglia that forms the dorsal or sensory roots. As the alar plates enlarge, dorsal median septum is formed. As discussed, the roof plate is the non-neural roof of the central canal which connects the two alar plates, and the floor plate is the non-neural floor of the central canal which connects the two basal plates. The floor plate contains the ventral white commissure. The outermost layer, the marginal zone, consists of axons arising from the neurons present in the mantle zone, forms the white matter of the spinal cord. The white matter, according to the direction of the fibers and functions, develops into three columns named as dorsal column, which is present between two dorsal gray horns, lateral column present between the respective dorsal and ventral gray horns, and the anterior column or ventral column present between the two ventral gray horns and anterior to the ventral gray horns. The dorsal column consists of purely ascending fibers or sensory fibers. The lateral and ventral columns have both ascending and descending fibers, that is both sensory and motor fibers. Development of spinal ganglia. The unipolar neurons in the spinal ganglia are derived from neural crest cells. The peripheral processes of the spinal ganglion cells pass in the spinal nerves to sensory endings in somatic or visceral structures. The central processes enter the spinal cord, constituting the dorsal roots of the spinal nerves. Development of spinal meninges. The mesenchyme surrounding the neural tube condenses to form the primordial meninges. The outer layer of this membrane gives rise to dura mater. The internal layer derived from the neural crest cells gives rise to leptomeninges, which includes pia and arachnoid matter. Fluid-filled spaces appear within the leptomeninges that soon coalesce to form the subarachnoid space. Cerebrospinal fluid starts to form during the 5th week of development. Myelination. Myelination of the spinal cord begins during 4th month in the ventral or motor roots. The sources of myelination is different in central nervous system and peripheral nervous system. Myelination in the central nervous system is brought about by oligodendrocytes. Myelination in the peripheral nervous system is brought about by the Schwann cells. Myelination of the corticospinal tracts is completed by the end of 2 years of age, and myelination of the fibers in association neocortex extends to 30 years of age. So that was the development of the spinal cord. Now coming to the development of rhombencephalon, it gives rise to two parts. Caudal part called myelencephalon and the rostral part called metencephalon. The myelencephalon developing from the caudal part of the rhombencephalon gives rise to medulla oblongata. In the spinal cord, we know the alar plate develops dorsal to the basal plate and or in the mantle zone surrounding the ependyma, and the central canal. In the development of medulla oblongata, sensory neuroblasts in the alar plate migrate from mantle zone into the marginal zone and form isolated areas of grey matter. As the pontine flexure forms, the rostral part of the myelencephalon widens and becomes flat, developing into upper part of medulla oblongata. The alar and basal plates, which was originally positioned dorsal and ventral, now changes its position in the cranial part of medulla oblongata. The alar plates are placed laterally and the basal plates are placed medially close to the midline. The central canal in the lower half of the medulla oblongata is present in the center as in the spinal cord. But in the upper part of medulla oblongata, the central canal is pushed to the dorsal surface and develops into fourth ventricle. Neuroblasts from alar plate form neurons arranged in 4 columns on each side from medial to lateral. General visceral afferent, special visceral afferent, general somatic afferent, and special somatic afferent. General visceral afferent, represented by the solitary nucleus, receives impulses from the viscera. Special visceral afferent, represented by the solitary nucleus, receives impulses from the taste fibers. General somatic afferent, represented by the spinal trigeminal nucleus, receives impulses from the surface of the head. Special somatic afferent, represented by the cochlear and vestibular nuclei present in the medullopontine junction, receives impulses from the ear. Dorsal column nuclei is represented by the gracile and cuneate nuclei. Inferior olivary nuclei, which are cerebellar relay nuclei, are derived from the alar plate by migrating to a ventral position. Neuroblasts from the basal plates form nuclei and organize into 3 cell columns from medial to lateral. General somatic efferent, special visceral efferent, general visceral efferent. General somatic efferent is represented by the neurons of the hypoglossal nerve and is called hypoglossal nuclei. Special visceral efferent is represented by neurons of cranial nerve 9, 10 and 11 and is called nucleus ambiguus. These fibers innervate the muscles derived from the pharyngeal arches. General visceral efferent is represented by neurons of the vagus and glossopharyngeal nerves and are called as the dorsal motor nucleus of the vagus and the inferior salivary nucleus respectively. You can observe the same details here with the diagram. You can observe that the basal plate is present on the medial side next to the midline and alar plate is present lateral to the basal plate. Neuroblasts from the basal plates form nuclei and organize into 3 cell columns on each side from medial to lateral. General somatic efferent, special visceral efferent, general visceral efferent. Neuroblasts from alar plate form neurons and are arranged in 4 columns on each side from medial to lateral. General visceral afferent and special visceral afferent are represented by the same column. Lateral to them is the general somatic afferent column and the most lateral is the special somatic afferent column. Now, let us identify the nuclei in each column which gives rise to specific cranial nerves from medial to lateral side of medulla oblongata. we will start identifying the nuclei from midline with basal or motor plate. General somatic efferent column is presented by the neurons of the hypoglossal nerve and is called hypoglossal nucleus. Special visceral efferent column is represented by neurons of the cranial nerve 9, that is glossopharyngeal nerve, cranial nerve 10, that is vagus nerve, and cranial nerve 11, that is accessory part of spinal accessory nerve and is called nucleus ambiguus. These fibers innervate the muscles derived from pharyngeal arches. General visceral efferent is represented by neurons of the vagus and glossopharyngeal nerves and are called as dorsal motor nucleus of the vagus and the inferior salivatory nucleus of glossopharyngeal nerve respectively. Coming to the alar plate, from medial to lateral, the nuclei are general visceral afferent column is represented by the solitary nucleus of facial nerve which receives impulses from the viscera. Special visceral afferent is again represented by the solitary nucleus of facial nerve which receives impulses from the taste fibers. General somatic afferent column is represented by the spinal nucleus of trigeminal nerve which receives impulses from the surface of the head. Special somatic afferent column is represented by the vestibular and the cochlear nuclei present in the medullopontine junction and receives impulses from the ear. When we take the cross-section of the adult medulla oblongata, it can be observed that all the nuclei derived from alar and basal plates are represented in the dorsal part of the medulla close to the fourth ventricle. Until now, we have discussed about the development of the nuclei derived from alar and basal plates of the neural tube. Now, we will see the development of the roof plate of the developing medulla oblongata. The roof plate of the developing medulla forms the lower part of the roof of the fourth ventricle. The roof plate is called the tela choroidea, which is a single layer of ependymal cells covered by pia mater. The tela choroidea is invaginated by the pial blood cells and both together form the choroid plexus of the fourth ventricle. The open part or rostral part or upper part of the medulla extends from the obex to the stria medullaris of the rhomboid fossa. The lateral walls of the rostral medulla open like a book and form the rhomboid fossa. That is the floor of the fourth ventricle. This opening of the upper part of medulla occurs due to the formation of the pontine flexure. As discussed earlier, the metencephalon developing from the rostral part or upper part of rhombencephalon gives rise to the pons and the cerebellum. The pons. Pons arises from the ventral part of the metencephalon. As in the upper part of the developing medulla oblongata, in myelencephalon, the pontine flexure causes the lateral walls of the pons to spread widely along with the grey matter in the floor of the fourth ventricle. The alar plate shifts lateral to the basal plate as in the medulla. The cavity of the metencephalon forms the superior part of the fourth ventricle. Now let us identify the nuclei in each column which gives rise to specific cranial nerves from medial to lateral side of pons. All the nuclei are present on the ventral part of the pons. We will start identifying the nuclei from midline with basal or motor plate. General somatic efferent column is represented by the neurons of the sixth cranial nerve and is called abducent nucleus. special visceral efferent column is represented by the neurons of the fifth cranial nerve, the trigeminal nerve, and the seventh cranial nerve, that is facial nerve, and are called facial nucleus and the motor nucleus of trigeminal nerve. General visceral efferent is represented by neurons of the seventh cranial nerve, that is facial nerve, and is called as superior salivatory nucleus. Now, let us identify the nuclei in each column of the alar plate, which gives rise to the specific cranial nerves from medial to lateral side of pons. General visceral afferent and special visceral afferent columns are both represented by one nucleus, that is solitary nucleus, or it is also called as the nucleus of solitary tract. This nucleus gives rise to seventh cranial nerve or facial nerve. General visceral afferent column, represented by the solitary nucleus, receives impulses from the viscera of head and neck. Special visceral afferent, again represented by the solitary nucleus, receives impulses from the taste fibers. General somatic afferent column is represented by three nuclei. The principal sensory nucleus of trigeminal nerve in the middle of the pons, spinal nucleus of trigeminal nerve extending caudally towards the medulla, and mesencephalic nucleus of trigeminal nerve which extends cranially towards midbrain. All the nuclei receive impulses from the surface of the head. Special somatic afferent column is represented by the vestibular and the cochlear nuclei present in the medullopontine junction and receives impulses from the ear. Both nuclei give rise to the eighth cranial nerve. When we take cross-section of the adult pons, it is divided into two parts, dorsal and ventral parts. The dorsal part is called as the tegmental part, and the ventral part is called as basal part or basilar part of the pons. All the nuclei of pons we discussed until now, which are derived by alar and basal plate, are present in the dorsal tegmental part. The basal or the ventral part of the pons contains the following structures: pontine nuclei, which consists of cerebellar relay nuclei and are derived from the alar plate by migrating to a ventral position. Corticospinal, corticobulbar and corticopontine fibers, whose cell bodies are located in the cerebral cortex, and pontocerebellar fibers. Coming to the development of cerebellum, because of the pontine flexure of the brain vesicles, the roof plate stretches on the dorsal side and the alar plate shifts to the lateral side of neural tube. The alar plates thicken dorsolaterally to form rhombic clips. These rhombic clips develop into cerebellum. The rest of the alar plate and basal plates develop into pons. The rhombic clips thicken at week 6 to form the cerebellar plate, which further grows and gives dumbbell appearance. When observed craniocaudally, the cerebellar plate is separated into cranial and caudal portions by a transverse groove. The cranial portion forms the vermis and the cerebellar hemispheres, both of which undergo extensive formation of fissures and folia. The caudal portion of the cerebellar plate forms the flocculonodular lobe, which is the most primitive part of the cerebellum. Like the rest of the neural tube, the rhombic clips consist of neurectoderm arranged in the ventricular zone, intermediate zone, and marginal zone. In the third month, the neurectoderm in the ventricular zone undergoes proliferation and forms the internal germinal layer. The internal germinal layer gives rise to the following structures: deep cerebellar nuclei, which includes dentate, emboliform, globose, and vestigial nuclei, Purkinje cells, and the Golgi cells. Some neuroectodermal cells from the internal germinal layer migrate through the marginal zone to form the external germinal layer. The external germinal layer gives rise to the following structures: basket cells, granule cells, and stellate cells. Both the external and internal germinal layers give rise to astrocytes, Bergman cells, and oligodendrocytes within the cerebellum. Development of midbrain. If you can recall formation of brain vesicles and its derivatives, the mesencephalon remains unchanged during the formation of secondary brain vesicle from primary brain vesicle. This unchanged mesencephalon give rise to the midbrain. The neuroblasts from the basal or motor plate of the midbrain give rise to the following cranial nerve nuclei. General somatic efferent column is represented by oculomotor nucleus of the third cranial nerve and trochlear nucleus of the fourth cranial nerve. Special visceral efferent column does not contribute to the formation of the cranial nerve nuclei in the midbrain. General visceral efferent column is represented by Edinger-Westphal nucleus of the third cranial nerve, then substantia nigra and the red nucleus. The neuroblasts from the alar or sensory plate of the midbrain give rise to the following cranial nerve nuclei. The general somatic afferent is represented by the mesencephalic nucleus of trigeminal nerve, which originates in metencephalon, that is the pons, and then migrates to midbrain. Note that the special visceral, general visceral, and special somatic afferent columns does not contribute to any nuclei in the midbrain. The alar plate also give rise to superior colliculi and the inferior colliculi. When we take cross-section of adult midbrain, it is divided into three parts: tectum, tegmentum, and crus cerebri from dorsal to ventral. The dorsal part is called as tectum. The middle part is called tegmentum, and the ventral part is called the crus cerebri of midbrain. All the nuclei of midbrain which are derived from alar plate and basal plate are present in the tectal and tegmental regions. Crus cerebri contain corticobulbar, corticospinal, and corticopontine fibers derived from the cerebral cortex of the telencephalon. Substantia nigra is present between the crus cerebri and the tegmentum of the midbrain. Development of diencephalon. Diencephalon develops from the prosencephalon, which is surrounding the walls of the third ventricle. The parts developing from diencephalon includes thalamus, hypothalamus, subthalamus, epithalamus, posterior lobe of pituitary gland, retina, ciliary body, and iris. As in other parts of developing neural tube, here also we see thickening of the middle layer, that is the mantle zone, forming the alar plates and basal plates. The alar plates remain prominent in the prosencephalon, but the basal plates regress. So, diencephalon is only the receiving center which passes on the information or sensations to the other parts of the brain for interpretation. The embryonic roof plate of third ventricle and dorsal parts of alar plates give rise to epithalamus. The epithalamus gives rise to several parts: the pineal body, habenular nuclei, habenular commissure, posterior commissure, tela choroidea, and the choroid plexus of the third ventricle. Alar plate also gives rise to thalamus, subthalamus, hypothalamus. Hypothalamus also develops from the floor plate ventral to hypothalamic sulcus. Thalamus includes the thalamic nuclei, lateral geniculate body, and medial geniculate body. The subthalamus includes the subthalamic nucleus, zona incerta, and lenticular and thalamic fasciculi, also called fields of Fortel. The subthalamus gives rise to neuroblasts that migrate into the white matter of telencephalon, developing into globus pallidus, which is a part of basal ganglion. The hypothalamus includes hypothalamic nuclei, mammillary bodies, and neurohypophysis, that is the posterior pituitary. Alar plate of diencephalon further gives rise to optic vesicles, cups, and stalks. The optic vesicles, cup, and stalk gives rise to retina, iris, ciliary body, optic nerve, optic chiasma, and optic tract. To know more about the development of eye, please watch the video on the development or embryology of eye. Development of hypophysis cerebri or pituitary gland. It is attached to the hypothalamus by the pituitary stalk and consists of the anterior lobe, also called adenohypophysis, and the posterior lobe called neurohypophysis. The location of the pituitary gland is present between the brain and the roof of the oral cavity. Pituitary gland develops from two processes. One process extending from the neurectoderm of the hypothalamus and the other from the ectoderm of the roof of the stomodium. The process which extends from the hypothalamus is called infundibular process, and one extending from stomodium is called Rathke's pouch. The Rathke's pouch, which is an ectodermal diverticulum of the primitive oral cavity or stomodium, develops into anterior lobe, pars intermedia, and the pars tuberalis. As the development proceeds, gradually the anterior lobe detaches from the stomodium and becomes attached to the posterior lobe. The infundibular process, which is a neuroectodermal ventral evagination of the hypothalamus, develops into the posterior lobe, infundibular stalk, and median eminence. Remnants of Rathke's pouch may give rise to a craniopharyngioma. A craniopharyngioma is the most common supratentorial tumor occurring in childhood and is the most common cause of hypopituitarism in children. Magnetic resonance imaging, that is MRI, shows a craniopharyngioma which lies suprasellar in the midline, compressing the optic chiasma and hypothalamus. So that was the development of the pituitary gland. Development of telencephalon. The telencephalon develops from the prosencephalon. Prosencephalon also includes the diencephalon, as already discussed. The telencephalon gives rise to the following parts: cerebral hemispheres, caudate, putamen, amygdaloid, claustrum, lamina terminalis, olfactory bulbs, and hippocampus. Cerebral hemispheres develop from telencephalon as bilateral evaginations of the lateral walls of the prosencephalic vesicle, which enlarges as it grows continuously. This continuous hemispheric growth gives rise to frontal, parietal, occipital, and temporal lobes, which overlie the insula and dorsal brainstem. The right and left cerebral hemispheres are interconnected by three commissures: the corpus callosum, anterior commissure, and hippocampal commissure. The surface of cerebral cortex changes gradually from a smooth surface or lissencephalic structure to a convoluted surface or gyrencephalic structure. As growth proceeds, a complex pattern of sulci or grooves and gyri or elevations develop. Cerebral cortex, like the rest of the neural tube, consists of neuroectoderm arranged in three layers: the ventricular zone, intermediate zone, and marginal zone. The neuroblasts from the ventricular zone and the intermediate or mantle zone migrate peripherally to marginal zone to form a transient layer called the cortical layer or the cortical plate. This develops into the cerebral cortex. The intermediate zone, that is the mantle zone, becomes devoid of neuroblasts and develops into white matter of the cerebral hemispheres. So that is the development of the telencephalon. With this, we have completed the 5 brain vesicles and its derivatives. Development of autonomic nervous system. The sympathetic nervous system originates from the basal plate of the neural tube, which is the ventrolateral thickening of mantle zone. The sympathetic system also develops from the neural crest cells. The basal plate of the neural tube gives rise to preganglionic sympathetic neurons within the intermediolateral cell column or lateral gray horn of the spinal cord. The neural crest cells gives rise to postganglionic sympathetic neurons within the sympathetic chain ganglia, prevertebral sympathetic ganglia such as celiac ganglia and chromaffin cells of adrenal medulla. The parasympathetic nervous system originates from the basal plate of the neural tube and the neural crest cells similar to that of sympathetic system. The basal plate of the neural tube gives rise to preganglionic parasympathetic neurons present within the nuclei of cranial nerve 3 in the midbrain, cranial nerve 7 in pons, cranial nerve 9-10 in medulla, and spinal cord at the level of 2nd to 4th sacral spinal segments. The neural crest cells give rise to postganglionic parasympathetic neurons present within the ciliary ganglion of cranial nerve 3, pterygopalatine ganglion of cranial nerve 7, submandibular ganglion of cranial nerve 7, otic ganglion of cranial nerve 9, enteric ganglia of the gut, that is Meissner and Auerbach's plexus, and cranial nerve 10, and abdominal or pelvic cavity and parasympathetic ganglia. With this, we complete the normal development of the central nervous system. Thank you for watching till the end. If you find this video useful, please like, share and comment.